Hello, thank you so much for the invitation. It's an honor for me to join you and share with you some of the research I've been doing. My presentation is titled, The Risk of Antimicrobial Resistance Pathogens, Case Study of Salmonella cerevisiae in Poultry Value Chains in Ghana. Here's an outline of my presentation. I'll start with an introduction, give some data on hazard identification and characterization, provide some information on poultry handling practices, exposure assessment, and risk characterization. Then I conclude. Salmonella is a significant pathogen globally. It is a bacteria that I think in sub-Saharan Africa is the most important. Non-typhoidal salmonella is commonly associated with all kinds of food, but particularly poultry. It causes diarrhea, sometimes bloody diarrhea, with fever and stomach cramps. Globally, it causes 93.8 million illnesses and 155,000 deaths each year. In Ghana, typhoidal salmonella is also common. It is associated with poor sanitation, but it can be transmitted through food and water. Some non-typhoidal salmonella are invasive. They get into the bloodstream and cause bloodstream infections with about 20 to 25% lethality. In a recent study in sub-Saharan Africa, Ghana recorded the highest prevalence of 63% of non-typhoidal salmonella in blood samples. Salmonella can be associated with diverse foods. Poultry has been identified as a significant product pathogen pathway, but other poultry products like turkey, guinea fowl, and duck that are also consumed in Ghana are significant, but chicken is the most common poultry product consumed. Salmonella has also been isolated from eggs in Ghana. It's associated with other meat products like pork and beef, and vegetables, green leafy vegetables like lettuce, amaranthus, cabbage, etc. at the farm level and at retail. It's also been associated with other processed foods like dairy products and other cereal blends that are used for children for complementary feeding. This slide shows a graph of the prevalence of Salmonella and other pathogens like Campylobacter and E. coli, but particularly Salmonella from raw chicken meat retail outlets in Accra. What's significant here is the green bar from the live bird markets. Live bird markets are wet markets where people get fresh chicken meat. We found that 77% of broiler meats we purchased from the live bird markets had Salmonella. From the cottage industries and the farm gates and the supermarkets, we found 42% prevalence of Salmonella in the chicken products. This table shows the different Salmonella cerevisiae we identified from chicken samples, but also from poultry environments. We found Salmonella typhimurium, Salmonella paratyphi B, Salmonella agona, Salmonella infantis, Salmonella Newport, Salmonella Mississippi, Salmonella Steffenberg, Salmonella Adelaide, Salmonella enteritidis, Salmonella West Hampton, and two other Salmonella variants that we could not identify the serovirus for, but we've sequenced these and we are yet to characterize them. Most of these serovirus were associated with the chicken carcasses, but a significant proportion were isolated from fecal matter, rinse water, and a few from bench surfaces, which shows opportunities for cross-contamination with other chicken carcasses. This slide shows antimicrobial resistance patterns of the Salmonella cereus from poultry environments in Accra. Out of the 55 isolates and exposure to 14 antibiotics, we found that most of these cereus were resistant to a lot of the antibiotics tested. However, the 4 antibiotics that are commonly used in invasive cases of salmonellosis or bloodstream infections, both for non-typhoidal salmonella and typhoidal salmonella, were quite sensitive. So in Ghana, we use a 3rd generation cephalosporins and fluoroquinolones such as ciprofloxacin, cefuroxime, cefotaxime, and chloramphenicol for treatment of invasive cases. Ciprofloxacin is a member of the fluoroquinolones, whereas cefuroxime and cefotaxime are part of the 3rd generation cephalosporins. These were, most of the isolates were sensitive to these antibiotics, so they remain good antibiotics for treatment of invasive cases. This slide shows the prevalence of multi-drug resistant isolates of Salmonella cereus. And you'd find here that most of the cereus we identified were multi-drug resistant to 5 to 6 and 7 to 9 antibiotics that we tested, which is very significant because we found that at the live bird markets and at some farm gates, they do dose the drinking water of the birds with antibiotics on a daily basis. So it's not surprising to see such high levels of multi-drug resistance among the isolates we found. This slide shows the chicken value chain and consumer food safety risk factors associated with purchase, storage, and handling at home. Most of the poultry products are obtained from the farm gate or from poultry farms. At the live bird markets, they purchase live birds from the farms and from all scales of farms, from backyard farming to highly industrialized farms. And they sell these birds at various traditional markets. Clients come in, choose the live bird they want, and if they require, the live bird market operators will slaughter and dress the live birds for them on demand. So they take their fresh chicken carcass home. But some farmers also process the birds and sell at the farm gate. We have grocery shops that also obtain live birds, slaughter and process at the grocery shops and sell the fresh chicken meat within the shops. Then we have a lot of cold stores that are spread throughout the country. You find them in every corner. They usually retail frozen chicken from imports, and some of the frozen chicken also end up in the grocery shops. A lot of Ghanaians obtain their fresh and frozen chicken from these outlets, and we found out that almost 88% of Ghanaians purchase their fresh chicken and frozen chicken and transport them at ambient temperatures home without chilling them or putting them in cooler bags. However, they do separate the chicken from other groceries they buy. At home, most people store their chicken either in freezers, about 88% of them, or in refrigerators. But in refrigerators, we found that about 89% of consumers stored chicken on the upper shelf, which presents a risk factor because the drips can contain salmonella and can cross-contaminate other products in the refrigerator. 93% of consumers store frozen chicken on bench top, which we do not encourage because on the bench top, it takes quite a while to thaw. And also as the chicken thaws and the temperature on the surface of the chicken increases to ambient temperatures, the Salmonella bacteria on the surface of chicken may have time to proliferate to high levels, which presents a risk factor for infection and cross-contamination. 93% of consumers wash their chicken before cooking. This is also discouraged, especially if washing is done under running tap water. This is because during washing, the Salmonella gets onto other surfaces, can contaminate the sink surfaces and benchtop surfaces, and provide opportunities for cross-contamination. 56% of consumers use same utensils for raw chicken and ready-to-eat foods. And out of these, only 45%, only 45% wash the utensils with soap and water in between use for raw chicken and ready-to-eat foods. These present significant hazard food safety risks that can cause cross-contamination events in domestic kitchens. Then we did some investigations at the live bird markets, and we interviewed live bird market operators on their food safety knowledge and practices. And we found out that 57% of the live bird market operators wash hands before slaughter and dressing of birds. A significant number of them, 43%, do not do this. 53% do not wash their hands with soap, they only wash with water. 71% do not wash their hands after handling chicken offal, and we know that the offal can contain a lot of Salmonella. 38% do not wash contact surfaces with soap and water prior to use, and 42% do not use disposable tissue when coughing or sneezing, nor wash hands afterwards. This study was, however, done before the COVID outbreak, and so we believe that a lot of these practices have improved afterwards, but it needs further investigation. This slide shows standard process flow diagram for processing of poultry for A, and B is the flow diagram for processing poultry at the live bird markets. As you'd see on this slide, there's no stunning of the birds, which is not a humane practice for slaughtering birds. There's slaughtering, bleeding, scalding, and defeathering, which is considered a critical control point. All the critical control points are in gray. Then there's singeing to remove the baby feathers. There's washing after defeathering and singeing. But at the live bird markets, water is a scarce resource, so the water is highly rationed. And most of the time, same water is used for multiple batches of chicken processing. Then there's evisceration, which is usually done by the experts. It's cut up, there's no chilling, there's packaging, and the client takes the product home. For the risk assessment, the objective was to estimate the level of consumer exposure to Salmonella from chicken consumption and characterize the risk of Salmonella infection from handling and consumption of chicken. The scope of the risk assessment was from slaughter to consumption of chicken. For methodology, we got a lot of data from our literature review. We also populated some primary data from our research project. Then we used Risk Ranger for risk profiling of Salmonella in raw chicken. Then we estimated the quantitative risk using at-risk software. This slide shows the model that we used for risk characterization and exposure assessment. First, we looked at retail and we considered Salmonella prevalence and measured Salmonella concentration. Then we looked at transport, where we considered time or duration of transport from retail point to home, the transportation temperature, and Salmonella growth rate during transport. And here we measured increased concentration of Salmonella due to transportation at ambient temperatures. Then we considered cooking. We looked at various cooking options, the duration of cooking, temperature of cooking, and Salmonella D values. And here we measured Salmonella inactivation due to heat treatment and Salmonella concentration after cooking. We also considered cross-contamination after cooking, where we looked at rate of transfer of Salmonella from other sources to cooked chicken, and we measured Salmonella levels after cross-contamination. Then for consumption, we looked at the quantity of chicken consumed and frequency of consumption, and we measured the quantity of Salmonella consumed. Table 4 shows results for the semi-quantitative estimation of the probability of illness from consumption of chicken contaminated with Salmonella. We used Risk Ranger for this assessment. Here, we determined the probability of illness per day per consumer to be 3.56 times 10 exponent minus 4. Total predicted illness per annum to be 3.8 exponent 5, and estimated risk at 59%, which is considered medium risk. So because we got medium risk, we decided to quantify the level of exposure of consumers to Salmonella and the illness, the number of illnesses per year from consumption of chicken. This table shows a summary of the input and output distributions from the quantitative microbial risk assessment on Salmonella from chicken. Our concentration at retail was a minimum of 0.6 log CFE per gram and a maximum of 1 log CFE per gram. Mean was 0.9. Concentration after transportation ranged from 0.6 to 2.5 log CFE per gram. Log reduction after cooking was about 10 to the 11 log reduction. Number of Salmonella cells after cross-contamination ranged from 0.06 to about 9 log CFU. Excuse me, sorry, 9 CFU, and amount of Salmonella consumed was estimated to be from 2.8 minimum to about 1,175 CFU. This table shows the estimated probability of illness from consumption of chicken contaminated with Salmonella. From the quantitative assessment, the probability of illness ranged from 4 exponent minus 5 to a maximum of 0.07 illness per person per serving. But the number of illnesses per person per year was estimated at a mean of 0.18, ranging from 0.004 to a maximum of 5.7 per person per year. The number of illnesses per 100,000 persons was estimated to range from 5 to 6,098, with a mean of 189 illnesses per 100,000 persons. So this indicates that Salmonella from chicken alone presents an appreciable level of risk to consumers. And this illness, if it's invasive Salmonella, could be very dangerous and could actually lead to a lethality. So measures need to be put in place to control Salmonella, especially at the farm gate and at retail. And we need to put out a lot of educational messages and create awareness on how consumers can properly handle chicken at home and prevent cross-contamination events. The level of risk can, however, be reduced when interventions such as biocide treatments using chlorine, brine, peracetic acid, vinegar treatments are employed. Proper hand washing after handling raw chicken should be promoted. Using separate utensils for raw chicken and other cooked or ready-to-eat foods should be encouraged. Transporting raw chicken under chilled conditions should also be encouraged. And we need to create awareness on the proper use of antibiotics at production levels and especially at the live bird markets, so that exposure of the birds to antibiotics is reduced and the spread of multi-drug resistant antibiotics are also minimized. I'd like to acknowledge the International Foundation for Science for funding this project and the entire project team, Dr. Patrick Njangi, Dr. Anthony Akunzule, various live bird market associations that supported our study, and Benjamin Ovi, Richard Ochwe, and Ms. Rebecca Ijimriku, who are students who worked on this project. And my special thanks goes to Society for Applied Microbiology for this opportunity. Thank you.